my channel. My name is Sanilal Sharma. So today I am going to discuss about the genetic code. So in this video we will discuss about genetic code, its feature and various things, how the genetic codes work. So let us discuss. Genetic code, it is the triplet sequence of nitrogenous bases that codes for a specific amino acid. We know that the nitrogenous bases are present in our DNA. There are large number of nitrogenous bases present randomly, and 3 of the nitrogenous bases, that can be anything, say for example A, G, A. 3 number of nitrogenous bases can form a single genetic code. So we will discuss how it works. So the main feature of the genetic code were worked during 1960. So in 1960 the main feature of the genetic code was done. Main feature were worked out. The scientists revealed all the characters of the genetic code. So let us know about the deciphering the code. So what is the meaning of deciphering? Deciphering means to decode, means the genetic codes are some codes, some codon, some sequence of nitrogenous bases. So knowing those codon, knowing about the function of the codon, what amino acid do they specially specify, all these things comes under deciphering the codon. So in deciphering there were various questions. Which codon specifies which amino acid? This was a question, that which codon specifies which kind of amino acid. How many of 64 codon are utilized? See, there are 64 codons. How it is formed, I will discuss. So there are 64 codons, and how many of them are utilized in specifying or coding a particular amino acid. How is code punctuated? That means, are the codon punctuated? Is there any kind of punctuation mark, or is there any kind of comma, full stop, stop or gap in between the codon? Then do they have same meaning in all the organism? So these were the questions by the scientist, and all those questions were answered by the scientist. So here I have given the name of some scientist, that is Marcel Nirenberg, Severo Oka, Hargobind Khurana, he is Indian, and Philip Leader. These were the scientists who worked on genetic code in 1960s. And in 1968 Robert Hawley, he also did various experiment and got Nobel Prize in physiology and medicine, and the Nobel Prize was shared by Hargobind Khurana and Marshall. So they 3 got the Nobel Prize in 1968. So let us know the function of the genetic code. See, before we start the function, you should know a little bit about the function of the DNA and the central dogma of molecular biology. How our DNA works. This is our DNA, which is going to form a protein. So to make a protein, the DNA will make a copy of it. So if this strand of the DNA contains C, 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 just for the convenience I am writing the same sequences, and if this contains G, 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 then any of the strand, that is the coding strand, that the strand which contains the code to form a particular kind of protein, that will be transcribed, that is called transcription, that I will discuss in my later video. It is a big process. In this process it forms mRNA, and this mRNA will be going carrying the nitrogenous base sequence, that is the codon, as it is in it, and it will be going to form a protein. If the mRNA is there, and say for example if I change the codon, like if I write it randomly like A, T, C, G, G, and in the opposite strand it will be T, A, G, C and C. So if this strand of the DNA was transcribed to mRNA, then that mRNA will be containing, in place of T there will be U, then exactly A, G, C, C. So like this the mRNA will be formed, and three of this codon, that is 3 of the nitrogenous bases, will be acting as a codon, and according to this codon present on the mRNA 3 the tRNA molecule will bring 1 by 1 amino acid. Now, it is a very big process. I am not going through all the details, but here I am explaining it a little bit. Like this DNA is inside the nucleus, and then it will form the mRNA, and then the mRNA will come out of the nucleus. It will go to the cytoplasm, and in cytoplasm it will go to the site of the protein synthesis. We call it ribosome, and in ribosome the mRNA goes and get fitted. Then this tRNA will take this 3 nitrogenous base as a codon, as a code, and according to that codon it will bring 1 amino acid. So, if for UAG it is of course a stop codon, but just to explain here, if for this codon there is an amino acid, say for example A, then A will be taken here by the tRNA molecule. tRNA have its anticodon part, here will be the amino acid, and in this end there is anticodon part. So, accordingly the anticodon part will contain some nitrogenous bases that will be the just complementary to the codon of the mRNA. It will go and it will sit here and it will give this amino acid, say for example A. For the next, tRNA will recognize the next 3 C. Accordingly, here will be G anticodon. It will go and sit here and it will give the another amino acid, say for example B. So, 1 by 1 amino acid will be linked according to this codon present on the mRNA. Now, very interesting thing is that whatever codon was present, whatever genetic sequence was present in DNA, the exact genetic sequence is present in the mRNA, and according to that order given by the DNA, the tRNA will recognize each codon 1 by 1 and will give the specific amino acid that the codon actually specifies. And when the specific number of amino acid and specific sequence of amino acid gets attached, it will be forming a polypeptide chain, and the polypeptide chain later forms a protein, and this process is called translation or protein synthesis. So, transcription, translation. So, here the genetic code plays a very important role. Say for example, in my previous video I have discussed about mutation. In mutation, what happens? There is a change in the genetic sequence, that is in gene mutation. So, what would happen if in place of A there is G, then here also in place of U there will be G, and if GAG, it was UAG, but now it has become GAG, definitely the amino acid will be changed here. It will not be A, say for example it will be Z. So, if the amino acid gets changed, if the sequence of the amino acid gets changed, then the polypeptide will be changed and the protein will be a different protein or a non-functional protein that will lead to the mutation. So, I have explained in my mutation video, you can watch it. So, here this is the function of the genetic code. So, genetic code are the triplet sequence of nitrogenous bases, and this triplet sequence of nitrogenous bases specifies a particular amino acid. So, this is the main character, main feature of the genetic code. So, there are different amino acid and they are specified by different codon. So, here I have an example. You can see this picture, that the first one is UUU phenylalanine, that specifies an amino acid, but the same amino acid can be specified by another codon, that is UUC. You can see stop codon, these are the stop codon UAA, UAG and UGA. These are the stop codon who do not specify for any kind of amino acid. Some of the codon can specify one amino acid, that is methionine, and that methionine cannot be specified or cannot be coded by another codon. So, here you can see this methionine contain AUG, and only AUG can recognize methionine, or only methionine can be recognized by the AUG, and methionine will not follow another codon, but you can see here in the last part arginine and arginine, there are 6 codon for the arginine. So this is a chart, you should know it. Okay, moving to the later part, let us discuss the property of the genetic code, and this is what the question is generally asked about. That is, number one property of the genetic code is they are composed of triplet nucleotide. So we all know they are always triplet, three nucleotide forms a codon, not the 2, not the 1, only 3 nucleotide are required. That is, total there are 4 nitrogenous bases: adenine, guanine, cytosine and thymine, and this 4, out of these 4, any of the 3, say for example A, C, C, it can be repeated. So this will recognize one amino acid. So genetic code is always triplet. This is one kind of character of the genetic code. Next, they are universal. Yes, genetic codes are universal. That means right from the bacteria till human and some other, that is actually the viruses, right from the bacteria, human and all the viruses, all of the organism, that is all the living thing, they have the same feature of genetic code. That means if a codon is there AAA, and if it recognize a particular kind of amino acid in case of human being, then this codon will be recognizing the same kind of amino acid in bacteria also. That is, through the course of evolution everything got changed, but the property of the genetic code is not changed in every organism, plant, animal, viruses, bacteria, fungi, everywhere their character is same. The same codon recognizes the same amino acid. Now let us move to the next character, that is they are non-overlapping. Non-overlapping means if there is a sequence of genetic code A, U, G, G, C, 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 G, U, A, then these 3 sequence of genetic code are there, and here the genetic code will be considered as first 3, then next 3, then next 3 and then next 3 and so on. That means one nitrogenous base will recognize only one codon. It won't be like AUG and in the next codon it will be recognized from UGG. No, it won't be happening, because if first 3 nitrogenous bases are recognized as the first codon, next codon will be recognized from the next nitrogenous bases. You cannot take this G or this U to make a new codon. That means you cannot overlap them. That's why they are non-overlapping. Next is they are commaless. So same thing, if there is A, G, G, C, U, C, A, C, U, G, G, randomly I am writing. If you see, the genetic codons are not having any kind of punctuation mark, like AGG is a first codon and there will be one comma, and the next will be this, there will be one comma, or there will be this next, there will be one comma. No, there is no comma, there are no full stop, there are no punctuation mark, there are no gap. They are just lying in a sequence, in a linear sequence, and the tRNA will recognize first 3, then the next 3, then the next 3, then the next 3, whatever it can be. If the DNA was already mutated and it formed a unrecognizable or unwanted codon sequence, then that unwanted or unrecognizable codon will be recognized by the tRNA, which will result in the mutation. There is one more thing, one more character, initiation codon. The genetic code have an initiation codon, that is AUG, and this codon always recognized for the amino acid, that is methionine. In all the organism, the protein synthesis will start from a first codon which will initiate the reaction, which will start the reaction, and to start the reaction the initiation codon is AUG. There is one more codon GUG which acts as initiation codon in case of some of the bacteria, but universally AUG is the first initiation codon. In every protein synthesis, the first codon will be AUG and the first amino acid will be methionine. But yes, if the methionine is not required in that protein, later the methionine will be cut out from the polypeptide chain, but to initiate the protein synthesis, the first initiation codon will be AUG. Next, the genetic code have stop codon or termination codon or nonsense codon. I don't prefer this name nonsense, but it is given in the book, but they have some sense. Why they are called stop codon or initiation or termination codon? Because when the protein synthesis goes on, one by one amino acid is given to the sequence of the mRNA according to the codon present on it, but this sequence of amino acid has to be stopped somewhere. It cannot be a long sequence of amino acid. So at a point where the amino acid requirement is over, when it is adequate, at that point the amino acid should be stopped. The binding of amino acid should be stopped. To stop that, at that point there must be any of the stop codon present, like UAA, UAG or UGA or UAG. If this codon presents at that place, say for example UAA, then this, out of this 3 codon anything can present, and if this present, it will not recognize any amino acid. So if it does not recognize amino acid, that means it is giving the signal to stop. Then the release factor comes and the protein synthesis stops here. So it has a very big role during the time of protein synthesis. So these are stop codon. They are UAA, UGA and UAG. They have some name: ochre, opal and amber. You should know it. Next is genetic code are degenerate. Degenerate means, you can see, like there are A, adenine, guanine, cytosine and thymine. In place of thymine you can say uracil, because we see the genetic code in case of mRNA. So out of these 4, if you make 3, if you take any 3 which can be repeated, so if you do that permutation combination, then you can get 64 codon. 64 codon. There are total 64 codon, and out of this 64 codon, 3 codons are nonsense codon or stop codon or termination codon. They won't specify for any kind of amino acid. That means how many codon left? There are only 61 codon left. This 61 codon will recognize those amino acid. And what is the number of the amino acid to form a particular protein? That is 20. 20 different types of amino acid are present in a protein. Yes, the number of amino acid can be 50 or 100 or 30. The number can be different, but the types of amino acid, only 20 types of amino acid are there. So if one amino acid is coded by one codon, then to code this 20 amino acid, there will be only 20 codon required. So still, out of this 61, if 20 codons are required to specify 20 amino acid, then still 41 codon left. What would they do? So they will also specify the same amino acid. That's why I show you in that image. You can see that image again, that one amino acid can be coded by many codon. That is called degeneracy of genetic code. And for example, I am given here, that is arginine can be coded by 6 codon. Only methionine have 1 codon, rest all the amino acid have more than 2 codon. That means if one codon comes, the amino acid A comes. If the degeneracy is there, then for the another codon the same amino acid can come at the same place. Next is they are non-ambiguous. The genetic codes are non-ambiguous. It means they are highly specific. One codon will always recognize a particular kind of amino acid. One codon will not recognize any other kind of amino acid. If AUG recognizes methionine, then later AUG will never recognize arginine. This never happens. It will always code code for methionine. That means whenever anywhere AUG comes, the methionine amino acid will come. This is specificity. They are highly specific. That is called, they are non-ambiguous. So there are some exceptions about the genetic code. So let us have some UAA and UGA. If you remember, these are the stop codon, but they specifies glutamine in case of paramecium. This is one exceptional case. And AGG, AGA, they act as stop codon in case of human mitochondria. So these are little bit of exception in genetic code. So I hope you understood the lecture. Thank you so much for watching. Thank you so much.